Good morning, everyone. Thank you for joining me this morning for Cherney College webinar series for pathogen detection platforms. I hope you all can hear me all right. If you can't, just let me know by saying something in chat. But we will go ahead and get started here. My name is Lauren Ellie. I'm the technical liaison here at Cherney Micro. This webinar is going to be recorded for others to view later. Please ask your questions, offer comments. That's what the Q&A and the chat is for. So please feel free to utilize those throughout the webinar. And there will be a Q&A, an open Q&A at the end of the webinar as well. So today we're going to be talking pathogen test methods. So I'm going to be going over the antigen-antibody interactions, which is alpha detection. Here at Cherney, we use the VIDAS platform, so I'll be covering that one. I'll also be covering the polymerase chain reaction method, also known as PCR detection. And here at Cherney, we use the IQ check prep, so I'll be going over that platform as well. So let's jump in with antigen-antibody interactions. So these types of interactions take place within the human body all the time. Let's back it up and define further what antigen and antibody means. So an antigen is any substance that causes your immune system to produce antibodies. For example, this can be chemical, bacteria toxins, viruses, or allergens. And an antibody is a large Y-shaped protein that is used by the immune system to identify and neutralize antigens. So you see in the picture there, depicted in green, is the Y-shaped antibody that will attach to that red antigen, and it's also sometimes referred to as a pathogen. So each antibody only binds to a specific antigen. So you'll also notice in that picture that on the antigen, there are different cell receptors, one's shaped more like a triangle and one's shaped more like a square. That antibody is specific to linking up with that portion of that antigen or pathogen. So in our bodies, this interaction is an immune response that, like I said earlier, can be caused by various bacteria, viruses, or even allergens. And the immune response creates memory and protects us from further infection in the future. But this interaction can also be used in other ways, like blood group determination, where they tell you whether you're blood type A, AB, or O. It can also be used in pathogen infection diagnosis, as well as ELISA testing, very similar to the alpha testing that I'll be discussing today, and immunofluorescence. So immunoassays can be designed for targeting certain antigen proteins and certain pathogens like salmonella or listeria. Antibodies or antigens are chemically linked with a detectable label. And these labels, they can emit radiation, they can produce a color change in a solution, they can also be fluorescent or be induced to emit light. So in the picture down below, you'll see in green, that would be our antigen. And the Y-shaped would be the antibody protein. And that little yellow marker coming off of that, that's going to be our detectable label that'll either produce that color change or produce a fluorescent light. So there are a couple different immunoassays. There is the ELISA and the alpha. So ELISA stands for enzyme-linked immunosorbent assay. Alpha stands for enzyme-linked fluorescent assay. So the alpha one is the one I'm going to be talking about, which generates that fluorescent signal that we learned about on the previous slide. There are three different types of assays here. There's a direct assay, where the triangle labeled AG, the yellow triangle on the bottom, is our antigen. The Y-shaped protein is our antibody. And then you'll notice that enzyme linked up at the top, which is going to be our fluorescent marker. So that's where the antibody is linking directly to the antigen, which is why it's called a direct assay. In the middle there, you have an indirect assay where your antigen is linked to a primary antibody, which is then linked to a secondary antibody that has that fluorescent marker on it. So it's indirectly, in a way, connected to that antigen, which is why we call it an indirect assay. The third type is a sandwich capture assay, where on the cell you've got a capture antibody and it's captured that antigen. So whether that's biological, virus, or even an allergen. So it's captured that antigen, and there has been a primary antibody that's attached and a secondary antibody with that fluorescent marker. So the sandwich assay is going to kind of sandwich that antigen in between different antibodies, and that's how its method of detection works. So here, like I said earlier, what we use at Cherney is the alpha detection, and for that we use our VIDAS platform. Our VIDAS kits come with specific kits for each pathogen, for instance, Salmonella or Listeria mono, etc. So for our VIDAS steps, our VIDAS is the alpha enzyme linked fluorescent assay. So first we're going to enrich that sample. Once it's done incubating, we're going to pull an aliquot or a small portion of the sample and put it into the strip that comes with the kit. We're going to heat it up, which is going to lyse the cells. It can be for 15 or 5 minutes, depending on the test or depending on which pathogen we're looking for. No matter what pathogen you are looking for, it is going to cool for 10 minutes at room temp. Then we're going to put it into the VIDAS machine and it'll run for relatively 1 hour. And then it'll interpret the result as either presumptive or negative. And so what's happening in that strip is, like on the right-hand side here, the cell in green, let's say, for example, it's a Salmonella pathogen. The antibody with the fluorescent tag is going to recognize that Salmonella pathogen and attach. And like I said earlier, those antibodies are specific to certain areas on that cell. So it's only going to attach to the A portion. It wouldn't fit with the B portion. So those antibodies are specific to certain areas on the antigen cell. So like I said earlier, VIDAS comes with pathogen-specific kits. Those kits come with individual strips for each sample, and each sample also gets what we call a spur, which is the cylindrical cone shape here. So you can see when I talked about pulling that small aliquot, that aliquot of sample goes in this first well, and the spur is going to act like a large straw, and it's going to suck up the sample, and then it's going to be able to distribute it through this pre-wash diluent and these various washing buffers, the conjugate, and it moves the sample through these different wells until it finally gets to the substrate one where it's able to read the fluorescent output, and it'll generate that into a either presumptive or a negative result. So looking a little bit closer on what happens there, the spur that I talked about that sucks up the sample is coated with antibodies specific for that particular pathogen. So if we keep using that Salmonella example, so if we were testing for Salmonella, those little red dots inside that well there, they are specific to the antibodies that are inside the spur that's going to suck up the sample like a straw. And those antigens, those Salmonella antigens, are going to be attached to those antibodies. And so it uses that sandwich assay that I had talked about on a few slides earlier, where that capture antibody inside the spur is going to grab the antigen, and then a labeled detection antibody with a fluorescent marker is going to light up. And by the time we reach the end of the strip, it's going to measure how many of those fluorescent antibodies have attached to an antigen, and it's going to be able to detect that and let us know whether it's presumptive or negative. So how does it do that? How does it know how much fluorescence means positive or negative? So that it does this by a certain threshold, and different test kits have different thresholds. So a negative result means it's below the threshold specified for that kit. A presumptive means it's equal to or above that threshold. So it expresses this in RLUs. So a Listeria genus, if it's less than 0.1, it'll consider it negative. If it's greater than or equal to 0.1, it'll consider it presumptive for Listeria genus. and so on and so forth throughout the table. They each have their own particular ones depending on which pathogen you're targeting. So that interpretation comes out to us here at Cherney as either negative or presumptive positive, and we'll let you guys know if you have a presumptive positive. And it's especially important for product that if you get that notification, you should always have those confirmed. Over on the right, there are some excerpts from AOAC and from the product inserts that state if you're using Listeria vitis, all positive results obtained must be confirmed. Likewise, below with the salmonella, it says your results should be confirmed because this is technically a screening method. So you'll want to go through the plate interpretation and make sure that you confirm those positive results. So a confirmation will result in either a positive or a negative. And what we'll do is we'll plate those on augers, either MOX, OX, XLD, or other chromogenic augers. And we're going to say, yes, typical colonies of either salmonella or Listeria are present here, and we'll say it's positive. Or we could also say, we don't see any typical colonies here and call that confirmation negative. Otherwise, you could also, if you have a positive confirmation, you can carry on with a speciation, which will tell you the genus and the species of the bacteria. For example, Listeria mono, Listeria being the genus, mono being the species. So that undergoes an identification process to determine that ID. And we will also confirm that with various biochemical identification and serological steps to confirm that we have found the right bacteria. So let's talk a little bit about things that can interfere with our VIDAS test. So residual sanitizer can affect your results of your VIDAS test. Neutralizing buffer, not to be confused with neutralizing broth. Over on the right-hand side, there are different types of neutralizing broths, DE, lethine, high cap, and there's also neutralizing buffer. So that one has been known to have some interactions with the VIDAS test and doesn't always work so well. So we recommend not utilizing that if you're sending in sponges to us for VIDAS environmental testing. I have a little smiley face next to high cap neutralizing broth because it is the one that we recommended. It works with various different microbiological testing platforms. So if you're looking for one, I would recommend the high cap. Other things that can interfere with the VIDAS test, sample matrix can definitely present some problems. The bacteria that's present, you could have some cross-reacting organisms in your sample that can cause kind of false reactions there. So the couple of those would be Citrobacter species, as well as Enterococcus that could give you different results where Citrobacter may look like salmonella and give you a presumptive. However, when we do that confirmation piece, we don't see typical colonies, and that could be due to a cross-reactor organism like Citrobacter. So let's switch gears and talk a little bit about PCR detection. PCR is a naturally occurring reaction used for amplifying DNA. This takes place in your cells when your cells are replicating. So we go from one parent strand of DNA, and it's then split into two strands, and those daughter strands are made by matching base pairs. So if you remember back to science class, talking about base DNA pairs, A pairs with T, and G pairs with C. So that's kind of that same process that we're using with PCR here. So in order to make that DNA split open during the PCR reaction, there are several cycles of heating and cooling, and that way copies of the target DNA, like our salmonella or Listeria, those amplicons are created through that process. So through PCR, there are specific probes that are used to detect the DNA during amplification. So these probes that I'm talking about are specific to the pathogen that you're looking for. So a probe, say if you're looking for salmonella, is going to be specific to salmonella DNA. So when that probe attaches to the salmonella DNA, it's going to be able to bind, and then it'll be able to complete the second strand of DNA. And it also has a fluorescent marker, much like we talked about with the antigen-antibody interaction. Once the probe attaches, it'll light up a fluorescent marker, and as amplification occurs over and over again, that fluorescent intensity is going to increase. And that's how we're going to detect whether it's presumptive positive or negative. So in the picture on the right-hand side, there's a single strand of DNA, and you'll also have that probe there with FAM is the fluorescent light that's going to go off once it attaches to the specific salmonella DNA. In this case, we were using salmonella as an example. And on the bottom, we see that the probe is not a match for that bottom strand. So there is no fluorescent signal that goes off, and there is no amplification. So like I said earlier, this PCR process is really similar to replication in your own cells. This process was developed in the 1980s and has since then become a very significant tool in molecular biology. So we take that one parent strand of DNA, unzips it into two strands, and then those base pairs complement each other as we create two new strands. Like I said earlier, A pairs with T, G pairs with C. So DNA replication in a thermocycler. So what this looks like is the picture on the right. We have on the top our double-stranded DNA. The denaturation step happens at about 95 C, where it's going to cleave those two DNA strands apart. So the next step, where it happens about 50 degrees C, the annealing is where those primers attach. So those primers that are specific to the salmonella or the listeria that we're looking for are going to attach to our DNA. It's going to increase the temperature slightly to 72 degrees Celsius, and those primers are going to be able to extend that DNA sequence to make two separate strands. And this cycles over and over again until we get an amplified amount of DNA that's detectable. So here's another way of looking at it. So at time point 0 over there, we've got one strand of DNA, and the purple line here is going to indicate an increase and decrease in temperature. So you'll see that we increase it to that 95 degrees and we get those strands to split apart. It's going to decrease it down to that number 2, where those primers specific to our pathogens are going to attach to those single strands of DNA if they're present. It's going to increase the temperature slightly, and it's going to be able to extend that DNA into two separate strands. So we went from one strand to two strand, and that was just in cycle 1. So it'll do that temperature cycle again in cycle 2, and there you go from 2 to 4. Cycle 3, you'll go from 4 to 8, and so on and so forth. It just keeps replicating at an exponential rate. So the key to this process is primers. You want those specific primers for the pathogens that you're looking for. So primers and probes are the starting point for PCR reaction. And it's going to recognize that specific sequence of target DNA in either your whatever pathogen you're looking for. So on the top of this target sequence, the primer there, let's say the primer we're looking for is salmonella detection. So that primer is not going to attach to that piece of DNA because that is not salmonella. So we won't have any primers attached. We won't have that extension or elongation. We won't have a separate DNA piece created from that. There's no detectable amplification. However, if you look at target sequence number 2 on the bottom, so we'll look at that specific piece of DNA, and that is salmonella, and it makes a perfect match. So as soon as that primer attaches, it's able to elongate that strand of DNA, and it's got that fluorescent probe on it, and it's going to keep amplifying, and that fluorescent marker will get stronger and stronger over time as those cycles keep going on and on. So real-time PCR is a specialized technique that allows a PCR reaction to be visualized in real time as those reactions progress. So as the number of cycles carries on, you can kind of visualize how that reaction intensifies for the fluorescent values. So real-time PCR allows us to measure minute amounts of DNA sequences in a sample. So to kind of step back and understand real-time PCR, let's imagine ourselves here with a PCR reaction tube at cycle number 25. So we're not starting out at cycle 1, we're starting out at cycle number 25 in that reaction tube. So what's in our tube at cycle 25? So we've got a mixture of DNA, we've got some primers, different enzymes, and at cycle 25, we've got, let's say, 1 million copies of amplicon DNA in that tube right now. So with real-time PCR, if we're looking at the graph, on the bottom is the cycle numbers, and on the left-hand side there, that's going to be the amount of DNA. So if we were looking at graphing this amount of DNA in our tube, from the very start until where we are right now at cycle 25, the graph would look like this. So you'll see that little exponential increase, where at cycle 25, we're at about 1 million copies of DNA. So what's it gonna look like after the next cycle, in cycle 26? Well, remember, this is an exponential reaction. So we started out at 25 with 1 million. So at cycle 26, with that replication process, we're gonna get about 2 million. Cycle 27, about 4 million. And in theory, at cycle 200, you would have tons of DNA, about 10 to the 35th power tons of DNA. So to put that in perspective, it would be the equivalent to the weight of 10 billion planets the size of Earth. And that clump of DNA doesn't exactly fit in our little PCR tube anymore. So realistically, as the chain reaction progresses here, it gets exponentially harder to find those primers and the nucleotides, and the polymerase that extends that DNA replicate is wearing out. So when we plot that real-time PCR, what it actually is going to end up looking like is that plateau at the top. So you'll see that exponential increase, but as we create more and more replicates of DNA, we're running out of primers in our solution that are able to detect and bind to salmonella because we've used them all up. So we reach some sort of a limit here once all of our primers are used up and we can't keep replicating DNA. So a very typical real-time PCR graph will always have that plateau at the top when you've reached your limit. So this is a very standard real-time PCR that allows us to look at the DNA amplification through the cycles. So like I said, here at Cherney, we use a Bio-Rad IQ check prep system. It runs the extraction using robotics. It's very handy. Our technicians back there love it because it's kind of a hands-off where the robot will do the work for you. It'll pipette out your samples, your reagents, your primers and probes and all of that. And once it's ready, it'll give you a completed PCR plate that you load into that thermocycler where you'll get those different cycles and the heat temperature rising and lowering. So with each round of amplification, the amount of DNA is going to increase, as does the intensity of the fluorescence. And the fluorescence is measured at each cycle, and the software plots the fluorescence against the number of cycles. So our process using a real-time PCR instrument consists of 3 main components. So you need the thermocycler, the PCR machine itself that's going to heat and cool, and the optical module, which is going to detect the fluorescence, those probes that are going to light up when they attach to the target DNA. And then you'll need the computer to translate the fluorescence into meaningful results. So when we're dealing with PCR, quality control is king. So the key quality control steps is going to be aseptic pipetting technique. So we're talking about DNA. You're not going to be able to see it. You're not going to be able to know whether you had some aseptic issues. So you really have to be concrete and have really solid aseptic pipetting techniques here. Another key quality control step is that temperature and time for heating and cooling. Your machine has to hit those different heat temperatures and cooling temperatures to allow for the DNA replication. Another key quality control step is cleaning, which also kind of goes along with amplicon monitoring. So you want to make sure that your PCR area is clean and you don't have any leftover DNA that could cross-contaminate other samples. So you're going to want to clean it with we use DNA away here, which is going to get rid of any residual DNA on your PCR surfaces. And then you're going to want to do routine amplicon monitoring to make sure that your cleaning that you're using is effective. Also, another key step is internal controls. So for every run, we have an internal control that makes sure that that time and temperature inside the machine work perfectly, and we had a positive control that was able to yield a positive result, as well as a negative control that yields a negative result. Some advantages and disadvantages to PCR. It's very high specificity, which is a great advantage. A low detection limit, 10 to the 2 CFU per mil. That real-time PCR, seeing that amplification and detection in one step is a great benefit. You get shorter assay times. Also, you can run simultaneous detection of multiple analytes at once. So you can do Salmonella and Listeria in the same run. Some disadvantages here: takes a lot of technical competence and skill. Like I mentioned, your pipetting has to be solid. You have to know how to prepare your reagents. You have to be wary of amplicon contamination. There's also a large cost for the equipment and price up front for those kits. So it might not be easy for labs starting out to make such a large purchase right away. There's also the issue with sample interference and inhibition. Some sample matrices will interfere with that fluorescent detection. So that can be one disadvantage. And that primer and probe design can be complicated for people to understand. So that's another disadvantage. It may also detect the presence of non-viable DNA. So this last one creates a much larger discussion, and I could talk about it all day. I suggest if you have questions about PCR and detection of non-viable DNA, reach out. It might be worth another webinar, but there's a lot to talk about on that one. So if you guys have questions, let me know. So things to consider when you're looking at both platforms, which one's right for you. Most importantly, you're going to want to know what testing is required of you by your customer or consumers. Some people say, you know, I need this test done via PCR, or some people aren't quite as stringent about it. So just make sure you know what's required of you. Also, it'd be good to ask your lab if your product type would be better suited to one platform over the other if you don't have those types of requirements set in place by your customers or consumers. But no matter which platform you utilize, know that both methods are excellent pathogen detection platforms that have been utilized for many years and produce quick, reliable results. Also, no matter what platform you're on, product presumptives on both platforms should always be followed up with that plate confirmation work that I stated earlier. So that concludes the webinar for today. I want to say thank you again for joining us. Please make sure to tune in for our other scheduled webinars: serial delusions and understanding results on June 2nd, and nutritional analysis on August 11th. And if you've missed any of our previous webinars or the 2020 webinars, they are available for viewing on our website. Also, I recommend you guys check out the blog section of our website for some more fun and educational discussion topics if you're looking for more knowledge.